In this video, we're going to be looking at a stop motion that is going to show DNA replication. So to start, we're actually going to use the enzyme helicase, and this is going to break the hydrogen bonds between the base pairs in your DNA molecule. It's going to make it into 2 strands. So let's watch this. So as you can see, that DNA molecule is being broken up into 2 strands via that hydrogen breaking. That's the main point you want to take away from this part. And your next step is going to be DNA polymerase. This gets a little complicated, but I'll try to put it in a nice condensed way for you. So basically, DNA polymerase is going to build complementary bases. So remember, helicase rips it apart. It separates the 2 strands, you could say. And then polymerase is going to build on those 2 separate strands. In this case, we have continuous movement from 5' to 3'. This is known as your leading strand in this example here. So we'll watch that. But another thing to note is that you might notice this little pattern that you're seeing on the DNA. Adenine will always go with thymine, A with T. Guanine will always go with cytosine, G with C. So as you can see, G, C, C, G, A, T. You can kind of guess what these ones are going to be, A, A. So that's the complementary base pair. I forgot to add that in just a little ago, but there we have it. So yeah, so let's watch the continuous movement from 5' to 3' in your leading strand. Remember, there's no gaps in these. So yes, just continuous movement, very fun. Now we have something a little complicated. This is going to be your discontinuous movement. This is going to be from 3' to 5'. Remember, DNA polymerase likes to go from 5' to 3'. Does not, if it goes backwards, it's going to have these fragments and gaps in them. And how are we going to fill that? We're going to fill that with ligase, but we'll get to that later. We'll get to that in just a second. So yes. So to recap, discontinuous movement, there's gaps in it. This is known as your lagging strand, I should say. And again, it's going to move from 3' to 5'. So let's hop into it. So as you can see, we're going to build a little bit, and then we're going to have a gap, some breathing room, and then we're going to have another segment built, another fragment, and then we're going to have a gap, and then another fragment. So we have these gaps, and how do we fill this? Well, if you remember what I just said a couple statements ago, we're going to use ligase to glue it together. So let's get into that. Boom. Filled. It's like glue. It's amazing. Ligase, you're amazing. And this is going to happen with all 3 of the gaps, or all 4 of the gaps, I should say. And what we're left with is 2 genetically identical DNA molecules. Remember, we started with 1. It was broken apart by helicase. DNA polymerase built some. Ligase glued the lagging strand together. And now we have 2 identical DNA molecules. But it's not what it seems. Although they are genetically the same, or on paper the same, visually the same, they are actually semi-conservative. And what does that mean? Well, we took DNA, 2 sides. So let's say we took this one and this one. These are your parent strands, and then you have daughter strands. And in DNA, well, we split it into 2, and then we built on. So we have 1 parent strand and 1 daughter strand for each DNA molecule that we replicated here. So we started with 2 parent strands. They branched off, and they built the daughter strands with them. So that's kind of what semi-conservative means. Hopefully I explained that pretty well. Trying to not get points off, so wish me luck, guys. But yeah, DNA replication is awesome, guys. Have a good day. Adios.